This is second lecture about the lac operon. In this lecture, we will be discuss about the how the exact lactose breakdown should be occur. So this is the bacterial cell. This is a bacteria. This is bacteria which contains lots of lactose in their surrounding. This pink color is the lactose which is present in the surrounding of the bacterial cell. So uh, this is lactose and this is bacteria, whole bacteria, and in the lactose present in the surrounding of the bacteria. Okay, and this is pink, this is green color, this green color is the permease enzyme. This permease enzyme will allow to enter the lactose inside the cell. Whenever permease enzyme allow to the lactose entered inside the cell, the lactose level should be high in the bacterial cell. And in the bacteria there are the DNA is present, genomic DNA. In the genomic DNA there are the operons are present, and here is the lac operon is present. The lac operon contains some structural gene, that is Z, Y and A. Z is responsible for beta galactosidase, Y is responsible for the permease, and the A is responsible for the transacetylase. So here is the operator and here is the promoter. In the operator there is the binding of the repressor or the activator depending on the situation, and depending on the presence of the lactose, the operator or repressor or activator should be bind. And in the promoter, the in the promoter side there should be the binding of the RNA polymerase for the transcription. So in short, this is the bacteria, and this bacteria contains lactose in their surrounding. And this permease, this green color is the permease, allow to enter lactose inside the bacteria, inside the bacterial cell. Whenever the lactose entered inside the bacterial cell, the lactose level should be high inside the bacterial cell, and this lactose should be bind to the repressor. This is repressor which is bind to the operator side. When there is the binding of the lactose into the repressor, this complex should be released. Whenever this complex should be released, there is the RNA polymerase should become. When this RNA polymerase, whenever this RNA polymerase should become, it bind to the promoter site. When it bind to the promoter site, there is the transcription and the translation. So in the transcription, in the translation, there is the mRNA produced. This is mRNA. From the mRNA, tRNA are produced, and from the tRNA there is the production of the rRNA, that is ribosomal RNA, and there is the protein synthesis. Which protein is synthesis? This structural protein is synthesis. This is ZYA. Z is responsible for beta galactosidase. Beta galactosidase is responsible for the breakdown of the lactose into glucose and the galactose, and lac-Y is responsible for the permease enzyme. So this is permease allowed to enter lactose inside the cell. And transacetylase, this thiogalactase transacetylase responsible to remove the toxic galactosidase out of the bacterial cell. So this whenever it totally depends on the presence or absence of the lactose. This is this is happen when there is the lots of lactose present into the bacterial cell, or present inside the bacteria. Whenever the lactose is present, the expression should be on. Whenever the lactose is absent, the expression should be off. So depending on the presence or absence of the lactose, the expression should be on or the off. Here the lactose is present, that's why mRNA is synthesis, and there is the production of the protein, and there is the binding of the RNA polymerase. So here the lactose is bind to the repressor site, and this complex is released. That's why RNA polymerase able to bind to the promoter sites, and there is the protein synthesis should be occur. This overall mechanism occur when there is the presence of the lactose. Let's see what happen when there is a not present lactose inside the bacterial cell. Here is not present lactose in the environment of the bacteria. So when there is a no lactose, there is no binding of the lactose to the repressor. When there is a no binding of the lactose to the repressor, so repressor is not released. So whenever the repressor is not released, there is a no binding of the RNA polymerase to the promoter site. Repressor role is to not allow to bind the RNA polymerase to the promoter site. Activator role is allowed to bind the RNA polymerase to the promoter site. And one more point here, whenever the lactose is bind, to the repressor, this complex, this repressor and lactose complex is slightly loose from the operator side. When it is loose from the operator side, this complex is released. So it totally depends on the lactose's presence or not. When lactose is not present, there is no binding of lactose to the repressor, so there is the no allowment of the RNA polymerase to come and bind to the promoter. And when there is the lactose is present, so there is a binding of the lactose to the repressor protein. When there is a binding of the lactose to the repressor protein, this complex is slightly loose and this complex is removed. So it totally depends, this whole mechanism is totally depends on the presence or absence of the lactose. So this is most important mechanism to understand the overall mechanism of the breakdown of the lactose. So when there is the production of this gene, this one, when there is production of the Z gene, Y gene and A gene, here is the mRNA synthesis. From the mRNA there is the from the mRNA synthesis there are the transfer to the tRNA. tRNA role is the transfer, and the rRNA synthesis. rRNA means protein synthesis. It occurs in the ribosome. So whenever this three protein is synthesis, there are the more uptake of the lactose inside the bacterial cell. When there is the more uptake of lactose inside the bacterial cell, there is the more breakdown of the lactose into glucose and galactose, and there is more amount of food available inside the bacterial cell. So this is overall mechanism, and it totally depend on the presence or absence of the lactose. And here is no presence of the lactose, here is no lactose in the bacterial cell, so there is no binding of the lactose to the repressor. When no binding of the lactose, there is the no RNA polymerase. When there is a binding of the repressor, so RNA polymerase will not able to come to the promoter site, and there is no expression should be occur. This is some general point about the mechanism. If the lactose is present, mechanism should be on. If the lactose is not present, mechanism should be off. It is in the and the tryptophan operon is completely opposite of the lac operon. So this is three main structural gene which is responsible for the breakdown of the and transportation of the lactose. The three structural gene are the LAC-Z, LAC-Y and LAC-A. Both produces the various enzymes. Beta galactosidase is responsible for breakdown of lactose into glucose and the galactose. So beta galactosidase is most important, and permease are responsible to transport the lactose, and transacetylase responsible to remove toxic galactosidase. The toxic galactosidase are removed by using the transacetylase enzyme. This is the reference which I refer for this overall process, that is Life Science Fundamental and the Practice by the Pranav Kumar and the Visha Meena. This is the book, best book to study the overall mechanism about the lac operon. Thank you.